must first apologize that I was not able to be present at this important meeting on the restoration of vision after visual system damage. It's an issue that I'm very interested in, and I think the recent progress in trying to find different means of restoring visual functions are very interesting and encouraging. My question and why I wanted to come here was to try to be part of this discussion and also present some of the evidence that Charles Gilbert and I have had in our work in the rhesus monkey, where we made retinal lesions and then in both eyes, so we could create a blind spot as if and then see what the effect would be on the cerebral cortex, the primary visual cortex of these retinal lesions, and see if it would be possible to restore functions. Because the visual system is a relatively clean system. We can make precise lesion and then study the effect of on the central part of the visual system. So let me first give a little bit of a background to the experiment. And I'm not, since I'm only 15 minutes at my disposal, I wanted to just briefly indicate some of the issues. As you know, the cells in the primary visual cortex are sensitive to orientation of contours, and this is a discovery that David Ubel and I made many years ago. And we also showed that the cell with the same orientation preference are organized in columns, as shown here in the first slide. Here you can see the cortical surface, white matter here, and we could show that if you make penetrations from the cortical surface with a microelectrode recording from single cells, and you first find the orientation of the first cell you record, let's say this is oriented around 2 o'clock, and you continue down in perpendicular to the surface of the cortex, you may stay and record from a whole series of cells with the same orientation preference. And this we call orientation columns. It's only layer 4 in the monkey where cells, like the incoming fibers from the lateral geniculate, have no orientation preference. We then could show, David Ubel and me, that these columns are organized in a very highly precise fashion, so that if you make an oblique penetration through the cortical superficial layers in the cortex, and here you start with a close to horizontal orientation of the optimum orientation for the cell, and you then have about 10-degree shift, about 18 shifts until you are back to the original orientation, the horizontal orientation. And each of these columns are about 60 or so micrometer wide. And so this is the sort of we like to say, the crystalline structure of the visual cortex. As you know, in addition, there are also ocular dominance columns, but this is not relevant for today's presentations. Another discovery that Charles Gilbert and I made later was that cells with the same orientation preference are interconnected. These columns of cell here have precise connections to cells over in this column, and they share then that they have the same orientation preference of a contour. And now the pathway for this is through what we call horizontal connections. Charles Gilbert and I discovered that here's a nerve cells, and you inject them with horseradish peroxidase, you find that the cell, the dendrites and the axons are filled in exquisite fashion, so you can follow them over several millimeters of length. Now, in this cell, if you rotate it 180 degrees, like if you look at my hand in this way and rotate it 180 degrees, you see that I have 5 fingers. And the same here, if you rotate this cell, you find that this cell have 3 fingers. And some cells go longer distances and you can have 5 fingers, as if. Now the fingers here are connections. These are the axons of these cells that we injected. And you can see this cluster around the cell body, but then it travels for about several millimeters to another cluster of connections here and still another here. And this, you can see that this cell also in the lower, below layer 4 here, have a cluster of connections which are in line with these. So this indicates that cells with the same orientation preference tend to be interconnected. And Charles Gilbert and I did a serious experiment actually with cross correlation and anatomical studies making it possible to demonstrate that this in fact is the case. And this led us to this model of how nerve cells are interconnected. So here you have a cell in the primary visual cortex, a pyramidal cell in the superficial layer, and this cell sends connections over to cells. This cell has a preference of vertical orientation. It sends an axon over to a group of columns of cell here with vertical orientation, and another group over here. Here we only have 4 different orientations indicated, or 5. But the point of this slide is to show that this is a very precise interconnection between the cell with the same functional properties that will, in this case, orientation preference. Now the question that Charles and I asked, what will happen if we eliminate this input, we destroy this input? This is a main input from the lateral geniculate body, from the thalamus, to the cortex, which is the main driving force of the cell in response to visual stimulation. What will happen if you eliminate this input? Can then the cells that send horizontal connection to the cell, can then they activate the cell? So that would mean that in order to stimulate this cell, you have its visual field, receptive field is going to be set aside from this, so you can actually stimulate this part without stimulating this. And you could, the idea was, if you stimulate out here, can we then drive this cell, which in turn would influence the firing of this cell? That was the question. And to carry out this experiment, we then made very precise laser with the laser lesions in the retina. Here's the fundus of the retina, and here the macula. You can see a very small lesion made here. And we made another lesion in the other eye on the corresponding point. So we would create a small blind spot in the visual cortex. In the retina also, obviously, because you destroy the retinal cells here. So this lesion area, there is it's not possible. The photoreceptors are dead. And it's important to remember that you have to do lesions in both eyes, because many of the cells in the primary visual cortex are driven both from the left eye and the right eye. In this case, let's just focus on the upper panel here, and this is we have a small area here of the cerebral cortex where we recorded about 15 or 16 cells by making several penetrations. And you can see here the receptive field of the small receptive field. These are cells with very specific orientation in this, organized in this topographical manner. Then in the laboratory we then made a lesion, and we made a marking for each site. We had made a recording, so we knew exactly the point where the And after the lesion, we then stimulated in the same area to see if we could activate the cells, and there were no response, because this is what you would expect since we have killed the receptors or the part of the retina that fed into these particular regions. Then we stimulated cells around to see could we in fact drive the cell from outside this region through the horizontal connection that I indicated before. Could we then affect these cells? And the answer was we could not. We could get no responses at all in this region by stimulating cells here, which should have horizontal, we know, have horizontal connection with these cells in this area. Now then we rented here for 1 year, and then we again, we have also done it for shorter times, as you can see below here, but just for the principle here, it's only important to remember that then we recorded from the cell, we had the photograph of the surface of the cortex, we had marked on this photograph the position where we had recorded cells, and as you can see, if we then recorded from a cell here, and we stimulate it out here, you could drive the cell. And this arrows here mean here is a big receptor field that you stimulated in this area here, you could drive the cell. Now the interesting part is that two things. One is that the receptor field was able to drive these cells was larger than the ones in here, as you can see overall. But the second very important part is that they were orientation selective, the driving here. So you have to stimulate in order to drive this cell in here that has lost its input from the retina, just to be absolutely sure that this cell has lost its input and only have input from here. And the finding is that as we stimulate out here with the right orientation, we can drive this cell. But only after a long time, you need several months of recovery of function. And and Charles Gilbert and Corinna Darian-Smith, postdoc in his lab, they later showed that these connections are actually not, they are sprouting. They had no evidence that new connections are branching and innervating cells, but the same connections are actually strengthened and sprouting, making the input to the cell more effective. And that presumably is a reason why it's now is possible to drive this cell from this input that otherwise, in normal case, input from here is so dominant, but now it's possible to make this weak input being sensed and effective to drive this cell. Of course, this cell's receptive field now will be out here, not here, because as you know, this input is feeding into here is gone. So it has been a rearrangement of but a functional recovery of the cell in the sense that it now responds to visual stimulation, and we believe that good evidence that this is through these horizontal connections that now has restored function. Now you can then think about the consequences of this just as an illustration to finish off, that here is then the retina, the original retinal lesion, which is still is a permanent lesion. It's the same even after a year. You have killed the retinal cells, receptors in this region. And initially you had a big scotoma, as it's called, a blind area in the visual cortex, since this was the case in both eyes. You had the lesion here in one eye and here in the other eye. And then after the cortex, this is a 2 months recovery, was enough to make it possible to drive the cells through horizontal connection. And then it's as if this scotoma is shrinking, because the cells now can respond to stimulation outside the original regions, and because you can see that the receptor fields here are larger, as you would expect. So that is the evidence that I wanted to present to you as sort of hard evidence that it is possible to restore function through time. Now in this case, we didn't make any special effort by stimulating the eyes in a similar way that Bernard Sabel has been doing in clinical cases with vision, trying to restore visual function. And it could be that if using special means, you could stimulate in the area around this area here, that you would get a quicker restoration of function or better. But nonetheless, it seems to me these kind of experiments would give you hope that there is more to learn from these kind of experiments, and also from the clinical work, that it should be possible to have patients restore vision in spite of initially apparent lack of vision. So the plasticity, therefore, that we are discussing here in the monkey was done in adolescent monkeys. But as we know from earlier work, plasticity in the monkey is pretty much over after 1 year, and these monkeys were older. So we can look upon this experiment as in the adult, similar to the experiment that Bernard has been doing in the clinic. And maybe we shouldn't be so surprised, because there are experiments from other laboratories like Bob Wirtz and Bill Newsom did many years ago, making lesion in the cerebral cortex, and showing that restoration of function actually after a small cortical lesion would occur quite surprisingly quickly. So this then is my contribution to this meeting, and I'm sorry not to be here to discuss this issue with you, and I was also honored to be chair of this session, and of course that is not possible. But I thank you for your attention, and I wish you a very good meeting. Thank you.